Hello, my name is Lindsay Hansen. I am a graduate student at Northern Arizona University in Environmental Science and Policy, and I'm also a graduate student at Miami University in biology. I did my undergrad at UC Berkeley, where I studied ecology and art, and I spent some time post-graduating working as a Watershed Stewards Program member at NOAA Southwest Fisheries Science Center, doing salmonid life cycle monitoring in coastal California creeks. Today, I'm going to be discussing the prevalence and concentration of microplastics in one of those California streams, looking at both the water column and juvenile salmonids. I worked very closely on this research with Rosalia Bond, Dr. Cynthia Kern, and Dr. Joseph Kiernan at the National Marine Fisheries Service Southwest Fisheries Science Center in Santa Cruz, California, in collaboration with UC Santa Cruz. They really made possible the research that I'm excited to share with you today. So some background. I'm sure you are all familiar with plastic, the notorious material that has struck quite a controversy as of late. It's cheap to make and easy to mold into various shapes and sizes per its name. However, single-use plastics are a major source of pollution, littering roadsides, beaches, and ocean gyres throughout the globe. Microplastics are plastic particles that are less than 5 millimeters in diameter. They are sourced from pre-production plastic pellets called nurdles. They're formed through the breakdown of larger plastic pieces, or they're manufactured directly as abrasives for personal cleaning products. Microplastics are proven to leach toxic chemicals when submerged in water and are vectors for invasive species and novel bacterial assemblages. Recent research has documented the ubiquitous nature of microplastics in the marine environment and the assimilation of microplastics in fishes at all trophic levels. Comparatively little is known, however, about the occurrence and concentration of microplastics in river ecosystems. The research I am discussing today is a first step in identifying the quantity and types of microplastic in smaller streams and within juvenile freshwater fish. The aim of this study was to quantify the concentration and characteristics, including the size, the shape, the color of microplastics in moving waters of Scott Creek, a small coastal California creek, as a starting point for assessing the environmental risk that plastic pollution has in small watershed systems. Now getting a little bit into our methods. The research was conducted in Scott Creek, a small 78 kilometers squared Central California coastal watershed, which is approximately 90 kilometers south of San Francisco. This creek is characterized by low flows in the summer, down to 0.1 cubic meters per second, and high flow periods in the winter and during isolated storm events, up to 65 meters per second. In the summer, a sandbar builds up at the mouth of the creek, closing off the creek from the ocean and forming a seasonal lagoon. In the winter, high flow breaks the sandbar and opens the creek to the ocean. The creek hosts sculpin, endangered tidewater goby, three-spined stickleback, threatened steelhead, and endangered coho salmon. And it's a site for ongoing monitoring and research on coho and steelhead populations. The lower watershed is sparsely populated, and dominant land uses include small-scale timber harvest, agriculture, while the headwaters of the creek are within the boundaries of Big Basin State Park, a park with an average attendance of around 800,000 annual visitors. We quantify the concentration of microplastics in Scott Creek in two ways. One, by sampling suspended microplastic in the water column, and two, by looking for plastics in the stomach contents of juvenile steelhead. To sample the concentration of microplastics in the water column, we sampled 5 sites along the main stem of Scott Creek, selected through stratified random sampling based on tributary inputs, with the idea being that different tributary inputs may bring a unique concentration of microplastics into the system. We used 3 stable manta nets with 363 nanometer mesh deployed in the water column for 30 minutes at each site. All microplastics found in the water column were 363 nanometers to 5 millimeters in diameter. While the nets were deployed, we determined the flow rate of water at the mouth of each net using a metal flow meter. Following the 30-minute collection period, we rinsed out all the material from the net using ambient site water into glass jars and stored them at room temperature. We repeated this process 4 times, once a month in April, May, June, and July, to reflect variability in flow from the high water spring to low flow summer. Back at the lab, we removed all particles, including stems, leaves, and twigs that were larger than 5 millimeters using stacked sieves, then dissolved all the remaining organic particles using ferrous sulfate heptahydrate and hydrogen peroxide reaction. Once the organic particles had been digested through this aqueous oxide reaction, we separated the microplastic particles from all other non-organic particles in a density separating funnel using salt water. The non-plastic particles were drained out of the funnel, and we used a vacuum filter to collect all remaining particles in the funnel onto a glass filter. To sample fish that had been in the creek their entire lives, we used incidental mortalities of juvenile steelhead from a down migrant trap set during the spring trapping season. For each fish, we excised the entire gastrointestinal tract, esophagus to anus, recorded fork length, and additional tagging information if present. We cut open the GI tract and removed all contents within using deionized water, collecting them on the same filter paper as the water samples. To account for potential ambient microplastics present due to our water sampling and laboratory procedures, we processed 6 water sample blanks, 3 from the water column, where we ran deionized water through the nets and continued the digestion process, and 3 lab blanks, which was where we ran DI water through the digestion process. Blanks were processed in a random order between water samples. Once all the samples had been sufficiently dried on filter paper, we examined each under a Leica S91I microscope using 6 to 55 times magnification. All microplastic particles were identified and quantified based on type and color. We removed all plastic particles from the sample and placed them on microscope slides. Results. This is an example of the sorts of microplastic found in our samples from Scott Creek. As you can see, microplastics under a microscope stand out from the bleached and dissolved organic material in texture, shape, color, and shininess. In all, we collected 60 individual water samples and 26 samples of steelhead gastrointestinal tracts, representing juvenile fish ranging from 44 millimeters to 155 millimeters. In both sample types, the majority of microplastics were fibers. In the water column, 91.2% of microplastics were fibers, while 97.7% of microplastics found in steelhead GI tracts were fibers. The remaining microplastics identified were fragments, shards, foam, and styrofoam. After standardizing for different volumes of water being sampled due to variable flow conditions over the study, we found higher microplastics per water volume during the low-flow July sample than in the high-flow samples from April and May. On average, water samples contained about 8 microplastics each, ranging between 0 to 22 particles per sample. Across all water samples, the most abundant fibers were black, about 43%, and dark blue, about 21%. We found 5 types of microplastics in our water column samples, including fibers, fragments, shards, foams, and styrofoam. Within these type categories, the microplastics present represented a wide array of colors and were likely sourced from various larger macroplastics in the watershed. The color and type of microplastics may be a clue to determine the sources of microplastic pollution in this creek. Of the juvenile steelhead sampled, 80.7% contained at least 1 particle, with a mean of 1.5 microplastics per individual fish. Some samples contained 0 microplastics, while the highest number in steelhead samples was 7. In steelhead, fibers made up 97.6% of microplastics found, with the most abundant fibers being black, about 11%, and dark blue, 39%. All of our blanks contained between 3 to 5 microplastic fibers, indicating possible environmental contamination. However, from our blanks, we learned that most ambient microplastic particles are short and mostly present as black or blue fibers and exist in low numbers per sample compared to the high concentration of microplastics present in each water sample. Most particles we found were secondary microplastics or macroplastics greater than 5 millimeters in diameter that had degraded into its present micro size. Examples of these include fibers, which are commonly sourced from runoff from laundry or the breakdown of materials like ropes, and fragments such as worn down pieces of tarps. Low flows had a higher concentration of microplastics compared to the lower concentration at high flows. It is likely that the higher flows dilute microplastic concentration in the creek. We also had expected low flows might lead to an underrepresentation of microplastics if the net was unable to fully float in the water. There may be an ambient level of microplastic, which was diluted in spring, or perhaps sources of microplastics are greater in the summer since the larger amount of sunlight during summer months may increase the breakdown rate of larger plastic pieces. We found very few microplastic particles in steelhead samples, especially when compared to blanks. This suggests that either juvenile steelhead in Scott Creek avoid consuming microplastics that exist in the water column, they pass microplastics through their gastrointestinal tracts quickly, or microplastics that they do consume are quickly incorporated into the fish's muscles and other organs, so they're not present in the gastrointestinal tract. Potential errors of this study include cross-contamination and identification biases. Because microplastic is such a ubiquitous material in air, water, and soil, the potential that our samples were cross-contaminated from other environmental microplastics exists. Furthermore, visual ID of microplastics can lead to overestimation of microplastic concentration. Microplastics less than 363 nanometers in size were not included in the study and may represent a greater threat due to their ability to translocate through tissues. Although it is a standard method, using an aqueous oxidizing reaction to dissolve organic material may cause microplastics to melt or become brittle, causing us not to be able to detect them in the microscope. Additionally, microplastics in fish can be incorporated into other tissues and muscles beyond the stomach, but we only looked at the digestive tract. In future studies, we suggest looking within musculature or within other organs such as the gills, which have direct contact with the water column. A suggestion for future microplastic research in streams is to use fine in-field water filtering methods, such as environmental DNA suction, to collect water samples. This method has the potential to collect more microplastic that might be too small to be caught by a net and could allow us to bypass the organic material digestion process, which may break down some microplastics. In conclusion, we examined the concentration of microplastics in the water column and juvenile steelhead tracts in Scott Creek, California. We found microplastics in almost all samples, mostly fibers and some fragments, shards, foam, and styrofoam, supporting our hypothesis that microplastics are present in freshwater systems, even in small rural creeks. The results of this study suggest that microplastic concentrations are higher in small creeks when flow is lower, but that concentration decreases with higher flows. We found very few microplastics in steelhead, which suggests that steelhead either do not ingest microplastics in Scott Creek, or they pass it through their gastrointestinal tracts quickly. We will continue to analyze this data to determine significant trends in microplastic concentration in Scott Creek based on location and flow conditions. I would like to thank the WDAFS 2019 Small Grant Program, which helped pay for laboratory materials and chemicals and tools. I would also like to thank my co-authors, Rosalia Bond, Cynthia Kern, and Joe Kiernan, and the Scott Creek Life Cycle Monitoring Team, Alexander Hay, Carly Liddy, Jeff Perez, and Joshua Miko for supporting this research. Additional thanks to California Polytechnic State University Swanton Pacific Ranch for providing land access, logistical support, and a secure place to conduct our research. NOAA Southwest Fisheries Science Center and UC Santa Cruz for tools and equipment, and Northern Arizona University for lab space. I would also like to thank Julie Masura from the University of Washington for advice with laboratory processing. Thank you for your interest. Here's my contact information, and please comment with any questions.